Yeah. Thank you so much, Seth, and Fabio and Peter and Ishwar for all of the coordination and everybody else who's been doing all this work to put on this huge, huge conference. So it's a delight to be here. And I'm very happy to talk with you all about a project and a topic that I've been interested in for several years now. And this is really about trying to understand the landscape of biodata resources worldwide. And so I just wanted to first say that in my position at the University of Illinois, I work with researchers across all disciplines. So arts, humanities, physical sciences, natural sciences. And I do have to say, generally speaking, it's amazing to talk with life scientists in general, just because they show such an extraordinary willingness to share and reuse data. And that truly is special and unique. And it's a really great testament to, the work that the community has done around that culture. Has created a different sort of challenge, though, I will say, in that there are just a lot of biodata resources then that are distributed throughout the world entirely. So basically anybody who has the data and, the motivation and, access to the technical resources can and does, establish their own biodata resource, which is wonderful in one perspective of thinking about, how great that data sharing is, but creates, a different cascade of challenges. So in particular, what I'm going to focus on today is really how do you find those resources to begin with, which has been actually a longstanding challenge in this area as well. So just starting a little bit to thinking about who's even looking. So certainly for the biodata resources, rightfully so, people think about their target audience first, who's actually directly coming to that resource to interact with the data. So these people usually have specific interests and are well connected to the community. So they're at conferences like this and reading, papers with their colleagues who are using those resources, et cetera. But there are several other sorts of audiences, which I call just sorts of spheres of interest. And so these are also groups that I tend to work with where we'll have somebody who's kind of on the periphery of a discipline or might be new or just interdisciplinary altogether. And so they're not quite as in the know or connected to a community that might have a resource that's core to it. So it's a little bit more difficult to think about how does a resource reach those people. And then the third category is the sort of general interest, and I think of this as sort of the observer of a discipline as a whole. So this could be kind of ecosystem surveyors, which I think of as myself in the context of this talk, but also ecosystem builders for those of you who are creating federated searches that are trying to gather data from lots and lots of different types of resource that might be all related to nematode research or some sort of topic. How do you even find the resources that you should be trying to connect to? So one of the things that's happened at the University of Illinois is I'll consult with people who are writing data management plans for grant applications. And in the life sciences, people will read, oh, I'm supposed to share data. That means I'm supposed to create a resource. And I was like, no. That's not, that's not what we mean by that. Ideally, there's a resource out there that you'll be able to deposit your data in already instead of establishing your own bespoke data resource. Though sometimes people be like, oh yeah, I know all about that, but I'm going to do it better. In which case I very diplomatically say, probably not. That's actually quite a bit of a bigger challenge than you think. And then also the sustainability of these resources is a huge challenge, as was, brought up a few times in the morning sessions. And I actually had this conversation with people so many times that I started to feel like I needed data myself to actually be able to show researchers that I was working with. So a few years ago, I did, I was very familiar with NAR and the database issue. So I decided to look through that as basically a historical sampling. So I went through all 3,000 articles that have been published in the database issue of NAR and did basically a census of all those data resources, if they're still available, if they're being maintained. And because I'm a open science advocate, I also shared all the code and the data for that. And for this audience in particular, I did want to point out this is an example of data that doesn't have a disciplinary data repository. It doesn't have a standard. It doesn't go in some specific place. And so it ends up in a generalist repository. And so I think sometimes people are like, what are those generalist repositories for? And it's actually for this kind of a thing. So it's this kind of specific one-off sort of data set that could have value. And in fact, it's been reused in a couple different ways. And in particular, the last was I was contacted by a couple of people that were in this kind of like ecosystem surveyor category that I talked about, in particular through ELIXIR. So I'm an American, I'm in the US, extremely jealous of ELIXIR because you look at that sort of effort and the effort to really coordinate the life sciences resources in a region, and we didn't have anything like that in the States, but I was obviously very aware that it's a global problem and certainly a problem in the US as well. And so one of the other people who contacted me was Chuck Cook, who's in the audience here. And this was just as the Global Biodata Coalition was just being launched. And so I was very excited to hear about this effort. So for those of you who don't know, or you might have seen, if you're at the morning sessions, a few people are at Core Biodata Resource, Core Resources. But this is an effort really to advocate for the essential nature of biodata resources and also to bring awareness around these perpetual threats to sustainability and to emphasize that there really needs to be global cooperation among the funders in order to address these sustainability issues. So Guy Cochran, who I'm not sure if is in the room right now, but is our executive director, and he will be giving a talk on Wednesday at the HFSP symposium, I think, which will include more on the Global Biodata Coalition. But just a few more words because it gives some important context for this talk. So this is a forum then for funders specifically to bring them together to really try to look at how to do the management and manage the growth and share strategies for this infrastructure as it continues to grow. And then in particular to ensure the sustainability of the financial support then for the core resources. So I'm going to be talking about a landscape analysis which includes thousands of resources. So the core ones are the ones that sort of underpin, a lot of the life sciences research and or the primary data source for some of the other resources. But they were particularly interested in this larger inventory than what I had done for NAR because one persistent challenge is that there's this lack of a clear understanding of the scope or the scale of this global biodata infrastructure. And so, and I've seen this in conversations myself where people would try to talk about sustainability and people just sort of throw up their hands and be like, well, we don't know how many there are or where they are, or we don't know anything about it. And so this is, one sort of an effort then to try to get at, well, okay, well, where are they and what are they? So the basic concept then was to create the most comprehensive inventory we could within a reasonable amount of time, and in particular using reproducible methodologies, partly because we're obviously very big advocates of open data and open science, but also because we knew ourselves that the inventory would need to be updated again in the future because the system is in flux. So this is quite a bit of a different setup than NAR, where I just knew right where to go. You go look at the database issue, and then it's a bit tedious, but and we used some text mining methods with that as well. But you don't know exactly the venue where everybody might be promoting their resource, and then of course you don't know the same thing, resource name or the resource locations. So if any of you are thinking like, oh, I hope they found my favorite resource, and it's like, you're cheating because you know what it is and you know exactly what it is. That's called a known item search. So that's a different kind of scenario. So of course our clues are similar though. So we're talking about something that's academic research related and data related, life science related, and online. So the basic strategy though was to look for papers that describe a biodata resource. And I'll talk more about the advantage of that and as throughout the talk. So starting with Europe PMC, which is a fabulous resource, very well organized, has a really great API, really great team to work with, and a really large, nice cache of open data. So that was another thing, is we really needed to start with open data if we wanted this to be reproducible and if we wanted people with variable resources throughout the world to be able to use this inventory. So starting with the API, we do a targeted search to get down to the PMIDs for articles that potentially describe a biodata resource, and then we use machine learning techniques, specifically BERT-based models for NLP, to first classify those articles about whether or not they do or do not describe a resource, and then extract the resource name using named entity recognition. So from there, we basically get a cursory list of PMIDs with the predicted resource names, and then we do some additional cleanup and augmentation, again going back to the Europe PMC API, because we can get additional information about the resource then, and for additional information about the resource to get the final inventory. So that additional information includes things like through author information, we can do or author affiliation information, we can do geolocating, and through funder metadata, we can get information about who's funding these resources. And then additionally, because it's using Europe PMC, there's SciLite annotations where text mining has already been done, so that's readily available. And then for any of the resources that publish in open access journal, then we have access to full text. And so then you become, the gates really flit open then in terms of that's a really, really rich data source for trying to figure out more information about the resource. In fact, a lot of times a richer information source than the website of the resource itself. I'll say that having looked at many hundreds or thousands of these over the years. So I'm not going to say a lot about anymore about the machine learning methods, but I just want to point out, give credit to our collaborators. So Anna and Ken. So Anna is at CCI and recently just did some really fabulous work on software mentions in the literature. And then Ken just graduated from the University of Arizona and has experience in bioinformatics and reproducibility in particular. So they did a lot of heavy lifting on this work. So just a little bit of overview of some of the results then. So we started with our targeted query, gave us 22,000 articles. The machine learning did the heavy lifting there. So classified about 4,300 those as describing a biodata resource. NER then was able to extract names for 4,000 resources. Then after that, we do a little bit of cleaning through URL extraction and some deduplication gives us 3,600 resources. And then at that point, we do a selective review to do a bit more disambiguation and also just check the lowest probability scores to try to make sure the inventory is as high quality as possible. So that gives us 3,112 resources in the final inventory then. So that's what we found. So this is over a 10-year period from articles from 2011 to 2021. So just a little bit of information then or results about that extra information that I said we could get through the article metadata. So first, of course, we looked at countries or funding we were particularly curious about, it being the GBCs. So as you might have guessed from this metadata, there's quite a long tail to this data. People will acknowledge anything from NIH as a funder to the local plant biochemistry department. So it's quite a long tail. So what we were really looking for was the most prevalent funders, and so there were 200 funders then who had funded at least 3 or more resources, which we found to be a nice good break-off there. And then we took those organizations and then mapped them to their country of origin. So, and the way the scale goes here is that the lighter color blue is the more resources associated with being funded by the organizations in that country. And then the darker blue is fewer resources, and gray we didn't get any data for. So for example, Argentina had 3 different organizations that were funding 6 different resources. So, but the big picture here is that you can automatically see like the global distribution of this infrastructure and the funding that's being contributed to this global infrastructure. So the second thing I'll show briefly then is just we did the same thing in terms of author affiliation. So we harvested out the countries for all the authors, and we did this through IP address as well. But authors, you want to think about that in terms of, we all know kind of intuitively that it's a kind of a constellation of people that are contributing to a resource, not necessarily just the location of that physical server. So we wanted to really think about that bigger network, and you can see immediately here that there's again quite a broader global distribution of people who are contributing to these resources at all as a whole. So this work is all available now. It's in GitHub, and we did quite a bit of work to try to make it reproducible. So there's a lot of documentation and different ways to kind of enter the pipeline and use different parts of it. So that's the code, data, and the documentation. And then we also put together a case study talking about our reproducibility efforts, which were fairly intense. And then we did a full paper as well. So those are out for peer review now, but they're available in Zenodo as preprints. So this is useful to GBC, from the get-go, thinking about the funders, because as a global organization wanting to make sure that there's good representation across the entire country, and it doesn't, or entire world, so you don't end up, just kind of being focused on certain geographical regions, which as social networks is really easy to accidentally do. So this is one way to be able to augment their efforts to really be a global enterprise. But there are a couple other ways that, we thought the inventory might be used. You can think of them a lot, actually, never enough time to do everything you want. But so, for example, in the United States, we've had what's known as the OSTP memos. There was one in 2013 and then another one just last fall or August. And these are basically directives from the White House of the government to the funding agencies about public access to research outcomes that were funded by taxpayers. And so the most recent one in August of 2022, again, emphasized that data sharing is an important activity for our researchers. So that means if researchers are supposed to share their data, then where are they supposed to share it? What are those resources? So this is, part of the reason why I've been so interested in it, because we also, again, don't want researchers just spawning even more resources that are then difficult to maintain. So this is actually how I was introduced to ISHWAR, is thinking about this topic overall. And NIH has a couple of different kind of internal lists of resources where deposit is allowed that they share with researchers and encourage them to consider for places to deposit their data. But they were wondering if there are additional resources that they had missed. So within this inventory, it certainly looks like there probably are some. So their lists usually have right around 100 resources, and we found well over 500 resources that were associated with NIH funding in the inventory. Now, we didn't, in this first kind of sweep of it, we didn't try to assess deposits or that sort of granularity in the inventory, but that's where, again, having articles that actually describe a resource are really valuable, because about 75% of those articles are open access. And so over 400 then of the resources of this, 566 were found via open access articles, which means we have access to that nice, rich, full text. So we were just doing this a couple of weeks ago. So just as a proof of concept then is we were just doing, text searches of, words you would expect to read if someone were able to deposit into a resource. So like upload or deposit. So this was a dead simple, like just text analysis at this point, but we already found a couple hundred. So as we, finesse that and make that better, that'll be a good way for people at NIH to basically prioritize which resources they'd want to take a look at to see if those are additional resources that could be promoted to NIH researchers for depositing their data. The last point too is about, well, what about articles that should have reported NIH funding but didn't? So about half, I said, have funding information, which is light years better than it was, 10 years ago. So we'll be grateful for that, but that still does mean there's a huge swath that are missing funding information. So one of the ways that we've explored just a little bit is like, well, we can take the PMIDs that don't have any funder information and actually check those against NIH's internal grant databases to see if there's actually a way to back in to finding out that a resource was actually funded by NIH. So if that feels like a wild goose chase, it is. So it's wildly annoying that we're doing all this kind of text mining and looking around and basically sniffing out, where the resources are. So these are all indirect methods, which then of course means that they are incomplete. And so this isn't what we want the future state to be by any means. Ideally, we would never really necessarily need to do a project like this. And this is, something that the community has also been aware of for, several years and has been working slowly, towards improving. So calling out a couple different efforts here. So there was the BioDB core, which was specification standards for databases that was published a number of years ago, specifically pointing out how useful these sorts of specifications and attributes are for people finding the resource to begin with. And then there was a 10 simple rules that was published a couple years ago too on developing databases. Rule number 9 was telling the world, which included publishing an article, and also my personal favorite, which is registration. So I was very happy to see a couple people earlier today did reference, their registration links in their talks. But there's really great resources like Fair Sharing and Re3Data, which are free ways for people to actually promote the resource. Word just needs to get out that they exist. It'd be wonderful then if we only had to look in a few locations to get access and find out about all the resources. And even in my work now, I use them a lot when I am looking for resources that somebody might want to deposit data in. But another thing that they do, that it actually prompts people to do something that's so simple, I find it embarrassing to even talk about it publicly like this, but is to actually name their resource and to use the name of the resource. So, Ken and I and Chuck to some extent too, did all the manual curation for the training data for the machine learning. And we read many abstracts where we couldn't really tell what the resource name. So it either wasn't present at all, or it was used so inconsistently, even within a little paragraph of an abstract, you weren't quite sure what the name of the resource really was. Or sometimes it would be indistinct, where there was other resources that were already named that, or sometimes just not very descriptive at all. Same thing with the URLs, make sure those are present, and a big shout out to the PID community for working on also trying to make sure that those are more persistent. So excellent work there. So when we talk about FAIR, we often talk about finding the data, the findability of the data within the resource, but I also think there's great work that can, continue to be done for finding the resource in and of itself. So, and even these simple things like, being really consistent and intentional about naming really improves the findability of the resource, which is good for all of those audiences, certainly the target users, but all these sort of periphery audiences as well. And then also for other discovery efforts. So this is one method that we used in order to get there, but there's certainly other ones as well. And I am almost out of time. So just want to thank my collaborators and CZI and the European PMC team who have been great to work with. And again, here's the access to the information that we have available.
Thank you, Heidi. So are there any questions in the room for Heidi? Anyone? Maybe we have 30 seconds for a burning question. Peter, do you see any?
There's a question. Yeah, there's a question in the room. Remember to say your name.
Yes, hi, Monique Tan from the SIB. I was kind of surprised and shocked to see that in your global breakdowns, there was nothing in Switzerland, which was just gray. So can you explain this?
Well, it just means, so in particular then for there's a couple of different things that can go wrong. There wasn't the data within the metadata fields, or it can go wrong in terms of like, for some reason, the Swiss name didn't match our algorithms for finding Swiss names. But probably, I would guess it's probably just more likely that the metadata, for whatever reason, what we didn't find just wasn't there. But I'll look at it more carefully and find out which of those things was actually true. Because, yeah, could be something like that.